Inside every cell in your body, there are roughly 3 billion base pairs of DNA. Printed out, they'd fill a library. Add in the millions of complex proteins folding and unfolding every second, and you have a massive, staggering amount of raw data running the human machine. For centuries, figuring out how that machine worked meant physically interacting with it. Scientists operated entirely in wet labs, environments where they manually handled liquids, mixing chemicals in petri dishes, and observed the physical results through microscopes. Every single test required physical matter and physical time. But this hands-on method has a hard limit. A human researcher looking through a lens cannot easily cross-reference a single genetic mutation against a database of 10 million other sequences. The human brain simply cannot spot microscopic disease patterns hidden across a dataset of that size. To solve biology, scientists had to treat it as an information science. By combining biological research with computer science, a field known as biotech computing, researchers let computers supply the massive processing power needed to make sense of all that raw genetic data. Biology is no longer restricted to physical liquids in glass tubes. It operates as a digital pipeline, taking biological matter as an input, processing the code, and delivering medical solutions as the output. Step 1 of this pipeline is the input phase. Before a computer can analyze a genetic disease, researchers have to extract the biological source code by physically reading or sequencing the raw DNA. Once that sequence is read, the genetic material is translated into giant digital libraries. These are known as biological databases, massive digital storage systems holding countless genome sequences and precise protein structures. Instead of freezing physical tissue samples, millions of genetic variations and medical records are stored digitally, where they can be instantly retrieved and compared. Digitizing the sequence means a researcher in Tokyo can analyze a DNA structure sequenced in London without ever touching a physical sample. This global data sharing entirely removes the bottleneck of physical transport and manual setup. But simply having the data sitting in a database does not cure a disease. To move into the processing phase, scientists need an immense amount of computational power to actually read and understand those billions of lines of code. They build specialized algorithms, step-by-step sets of instructions that tell the computer exactly what to look for. These software engines rapidly scan billions of letters in the digitized DNA sequence. The algorithm hunts for hidden anomalies. It searches for a single typo, a specific mutation causing a cellular malfunction, leading to infection, genetic disorders, or cancer. To find that one specific error among 3 billion base pairs, a human would have to spend a lifetime manually aligning and comparing sequences. A computer cluster runs that exact same comparison in a matter of seconds. These algorithms act as the ultimate diagnostic tool. They process raw, chaotic strings of biological data directly into clear, actionable medical intelligence. Once the problem is identified, the pipeline moves to the final stage, output. Before a new chemical compound is ever given to a human patient, the proposed medicine is tested in a highly controlled virtual sandbox. Computers map out predicted biological behaviors. A key focus is protein folding, the physical process where long chains of amino acids lock into specific shapes to function properly. The simulation calculates exactly how a new drug will interact with those complex, three-dimensional structures. Testing 1,000 different chemical compounds in a physical lab takes years of trial and error and costs billions of dollars. Running those same interactions through a digital simulation narrows down the viable options in days, drastically lowering the cost of research and entirely removing the risk of harming human trial subjects with toxic compounds. By validating treatments digitally first, medical science moves away from reactive physical trial and error. It becomes a process of proactive, predictive biological engineering. We saw the immediate global impact of this processing power during recent viral outbreaks. By tracking virus mutations through data analysis in real time, researchers were able to develop targeted vaccines in months rather than decades. At the clinical level, this pipeline enables personalized medicine. Because a doctor can now sequence your specific genome, treatments can be customized to match your exact DNA profile. This maximizes the effectiveness of the drug while minimizing adverse side effects. Now, the pipeline is absorbing its most powerful tool yet, artificial intelligence and machine learning. Machine learning is a type of computer science that allows systems to learn from data and improve their own accuracy without being explicitly programmed. AI shifts the pipeline from simple retrieval to active prediction. These systems analyze patterns across millions of genomes to forecast how a biological system behaves under new conditions, allowing researchers to design entirely original protein structures to target a specific disease. We have graduated from reading the manual of life to actively rewriting its pages. By editing genetic material based on these precise models, modern science has transitioned into a phase where the cure for an individual patient is computed before it is ever manufactured.
We are about to reach 100 subscribers. Please like, share, and subscribe to complete the target.